I thought it would be interesting to discuss CAR-T cells in the context of B-ALL therapy. I'll go over the limitations and risks of this potent immuno-oncologic therapy and what I think is an interesting application of CRISPR technology in this space. So let's begin with a background on B-ALL. B-lymphoblastic leukemia/lymphoma is a neoplasm of precursor lymphoid cells committed to the B-cell lineage. These cells typically are composed of small to medium-sized blasts with condensed chromatin, inconspicuous nucleoli, and scant cytoplasm. They can involve the bone marrow and blood, referred to as B-ALL, or can occasionally present with primary involvement of nodal or extranodal sites, in which case we call it LBL. Unlike with myeloid leukemias, there is no agreed-upon minimum blast count required for the diagnosis, but in general, the diagnosis is avoided when blasts are less than 20%. It is primarily a disease of children. About 75% of cases occur at age 6 or younger. B-LBL constitutes about 10% of lymphoblastic lymphomas, with the remaining being of T-cell lineage. There are currently 9 genetically defined B-ALLs, according to the 4th edition of the WHO. The 922 BCR-ABL1 fusion, more common in adults, about 25% of cases, versus children, where it's seen in about 2 to 3%. And in these cases, the blasts are associated with CD25 expression. KMT2A rearrangements are most commonly found in infants less than 1 year of age. Typically, they will be CD10 negative. It's important to look for monoblasts, which may signal a B-myeloid acute leukemia. ETV6 RUNX1 fusion is common in children, about 25% of cases, never seen in infants. It's a very favorable prognosis with complete response in over 90%. CD20 and CD9 are frequently absent in the blasts, and cryptic translocations will require FISH to identify this fusion. B-ALL with hyperdiploidy. This is more common in children, very rare in adults. It's defined as blasts containing over 50 chromosomes, usually without translocations or other structural alterations. B-ALL with hypodiploidy is quite rare, less than 5% of all B-ALL cases. It's seen in children and adults and has a poor prognosis. Hypodiploidy is separated into 3 classes. Near hypodiploidy often has RAS or receptor tyrosine kinase mutations and the worst prognosis. Low hypodiploidy is associated with TP53 mutation and Li-Fraumeni syndrome, and high hypodiploidy. IgH-IL-3 fusions are very rare, less than 1% of all cases, and are associated with reactive eosinophilia and possibly have a neutral prognosis. Blast counts may be deceptively low in these cases. TCF3-PBX1 is common in children, about 6% of cases, less common in adults. Typically, the blast will lack CD34 in these cases and has a favorable prognosis with treatment. BCR-ABL1-like is a provisional entity in the 4th edition of the WHO. It's relatively common, occurring in about 10 to 25% of patients with ALL, and increased frequency with age. These cases have translocations involving other tyrosine kinases like CRLF2 in about 50% of cases, which is also a useful flow marker in such cases. Or, less commonly, truncation and activation of EPOR. This entity has poor prognosis and higher post-induction MRD rates. And finally, IAMP21, amplification of a portion of chromosome 21. More commonly seen in children, about 2% of cases, and is detected by a FISH probe for RUNX1, which shows more than 5 copies. It's associated with the Robertsonian translocation 1521, which has a 3,000-fold risk of developing ALL if found, and relatively poor prognosis without treatment. Treatment is categorized in 2 basic subsets: Philadelphia positive versus Philadelphia negative, then further subdivided based on patient age and risk stratification. Here's a simplified breakdown of the treatment regimens. Treatments are, in general, similar, with the addition of TKI in cases of Philadelphia positive ALL. Blinatumomab is a bispecific T-cell engager. It binds CD3 and CD19, bringing T cells and B cells together and activating the T cell. Inotuzumab ozogamicin is an anti-CD22 antibody that carries the cytotoxic molecule ozogamicin, which internalizes and damages the DNA. Busatinib is a second TKI used when there is resistance or intolerance to another TKI. Tisagenlecleucel is a second-generation anti-CD19 CAR-T therapy, and it is not considered until refractory disease in patients less than 26 years of age or after 2 or more relapses at any age. So what is a CAR? It stands for Chimeric Antigen Receptor, and as its name implies, it is a receptor made from different protein motifs and domains that is designed to bind an antigen of our choosing. The purpose of this synthetic receptor is to recognize a particular antigen on a target cell and activate an immunologic response against that target. That immunologic response comes from the T cells in which we engineer to express our synthetic receptor. This allows us to bypass the MHC-directed pathway of T cell activation and provides a tumor-specific attack. Tisagenlecleucel, the anti-CD19 CAR-T therapy, was developed by Novartis and is the first gene therapy-derived treatment to receive FDA approval in 2017. The 4 main components of a chimeric antigen receptor are shown here. The antigen binding domain is the business end of the CAR receptor. There are 2 characteristics of the binding domain that are important: affinity and specificity. Specificity is clear. You want the binding domain to recognize a specific epitope on tumor cells to avoid off-target effects. The affinity of the receptor is also an important parameter that determines CAR function. There should be high enough affinity that the CAR receptor activates the T cell, but if the affinity is too high, it can actually result in activation-induced death of the CAR T cell and trigger toxicities. Affinity also relies on the number of tumor antigens available. For instance, if a tumor expresses abundant CAR antigens, having a lower affinity might be preferred to avoid toxicity. But too low, and you fail to meet the minimum threshold required to activate the T cell. The hinge or spacer region provides flexibility to overcome steric hindrances and allow the antigen binding domain to access the targeted epitope. The length and composition of the hinge region can affect flexibility, CAR expression, signaling, epitope recognition, and the strength of activation outputs. The proper length is often tailored for each specific antigen binding domain. The anti-CD19 CARs have short spacer regions. The major function of the transmembrane domain is to anchor the CAR to the T cell membrane. This domain is the least characterized of all the CAR domains, and there is some evidence suggesting it may influence CAR expression level, stability, and even signal activation. An example of this is the use of CD3 zeta transmembrane sequences. This transmembrane domain facilitates T cell activation by mediating CAR dimerization, but comes at the cost of stability compared to other transmembrane domain sequences. Another example is the use of the CD8 alpha transmembrane domain, which negatively influences the release of TNF and interferon gamma by the T cell, but also has decreased susceptibility to activation-induced cell death compared to other CAR domains. And finally, the intracellular signaling domain. A lot of focus has been placed on the intracellular domain in an effort to optimize CAR T cell function. First-generation CARs, engineered from the late 90s, contain tyrosine-based activation motifs from CD3 zeta. Unfortunately, they were not effective enough by themselves to generate an appropriate T-cell response, and clinical studies showed limited to no efficacy. A co-stimulatory domain is needed to maintain T-cell activation, and this prompted the second generation of CARs, which added the co-stimulatory domain in series with the intracellular activating domain. The second generation CAR-T cells produced strong therapeutic responses in several hematologic malignancies, including CLL/SLL, B-ALL, DLBCL, and plasma cell myeloma. And the efficacy of second generation CAR-T cells are being investigated in solid tumors now as well. The derivation of third generation CARs, which incorporate two co-stimulatory domains, was brought about because it was thought that a single co-stimulatory domain may result in incomplete T-cell activation. However, preclinical studies with third generation CARs have produced mixed results to date. How do we get T-cells, and how do we engineer them to express our synthetic receptor? T-cells are isolated from either the patient for an autologous transplant or from a matched donor for allogeneic transplant. Autologous transplant efficiency is actually lower simply because patients tend to be on chemotherapy regimens that decrease their overall cell counts. Once acquired, T-cells are then isolated from the other hematopoietic cells and cultured in vitro. The process of isolating T-cells, engineering them, and producing a viable product ready for infusion requires about 20 days and can really only be done at special centers with the appropriately trained staff. Now, generating CAR-T cells requires us to insert our CAR gene into the genome of T cells. The most common method of gene insertion relies on lentiviral vectors, which are derived from HIV sequences that are redesigned and reprogrammed to insert our gene of interest. These are highly efficient gene transfer vehicles and produce stable expression of the CAR gene. The biggest worry with using lentivirus is insertional mutagenesis. Lentiviral vectors insert the gene of interest randomly in the genome, and there is always a concern that this could result in oncogenesis. Tisagen-leclucil is produced with lentiviral vectors and has had to undergo extensive biosafety studies for FDA clearance. But the concerns and risks for oncogenesis still remain. To date, the issues surrounding CAR-T cell therapy revolve around the mechanistic aspects of the therapy and not with the lentiviral derivation. There are no reports of lentiviral-driven oncogenesis in CAR-T cell recipients. Lastly, while lentiviral integrated genes produce stable expression within a cell, there is considerable variability in the copy numbers among a batch of T cells. We simply cannot control how much lentivirus infects a single cell. So some may receive few copies of the CAR gene, others may receive several. And this ultimately results in varied potency and toxicity potential among the batch of T cells. Zinc finger and talons are older gene editing techniques that have been used in creating CAR-T cells, but they suffer from low integration efficiency, technically labor-intensive methods, and are harder to scale up for mass use. In the last few years, there has been increased discussion and hype around CRISPR-Cas9, and some of it is justified. Almost as soon as news about this tool hit mainstream, every gene editing lab has wanted to get their hands on it, and it is currently being used in numerous clinical trials, ranging from inherited disorders like hemoglobinopathies to cell-based therapies, including CAR-T. So what is it? CRISPR stands for Clustered Regularly Interspersed Short Palindromic Repeats. It describes a family of DNA sequences found within the genomes of bacteria and archaea that function as an acquired immune system. These sequences are derived from DNA fragments of viruses that have previously infected the prokaryote and are used to detect and destroy DNA from similar viruses during a subsequent infection. Cas9 is the CRISPR-associated protein 9, and its function is as an endonuclease that uses CRISPR sequences as a guide to recognize and cleave specific strands of DNA from a viral infection. The greatest advantage of this whole system is its precision. CRISPR-Cas9 allows gene editing at a single nucleotide resolution within a sequence of our choosing with high efficiency, something that cannot be achieved with lentiviruses or other gene editing tools. And the technical aspects are easy to implement, another great advantage to the prior methods. It's also scalable, which is something Zinkfinger and Talon struggled with. There are recent reports in the literature, however, that show CRISPR-Cas9 may have off-target effects elsewhere in the genome, and this is still being looked at, so cautious optimism is perhaps the best approach here. So how does CRISPR actually work, and how do we use it for our own designs and purposes? The Cas9 enzyme combines with a guide RNA that we designed to target a specific sequence in the genome, and this forms the Cas9 complex. The complex reads along the genome, stopping at specific regions known as protospacer adjacent motifs, or PAM. If there is sequence homology between the guide RNA and genome sequence at one of these stops, the Cas9 enzyme activates its endonucleases to cleave both strands of DNA. Now, repairing double-stranded DNA breaks through non-homologous end joining is extremely error-prone and introduces random nucleotides to repair the break. If this occurs within a gene, it can disrupt the gene completely. We can modify the Cas9 enzyme to do other things besides cleave DNA. In this case, a transmutase is attached which allows the Cas9 complex to induce a point mutation at a specific site we want. Other modifications exist that allow us to promote or repress gene expression. There are even modifications that attach fluorescent proteins to the Cas9 complex that allow us to study the three-dimensional architecture of the chromosome. And more recently, we are able to even introduce sequences of our own in a more targeted and controlled fashion, unlike the random insertions and variable copy numbers seen with lentiviral vectors. How is this being used to engineer CAR-T cells? CRISPR Therapeutics is currently undergoing phase 1 trials for their lead immuno-oncology product, CTX110. This is a CRISPR-derived anti-CD19 CAR-T cell engineered in a way that they hope will allow faster, off-the-shelf use. They perform 3 genetic modifications of the T cells using CRISPR. Gene knockout of the TCR and MHC by non-homologous end joining, and knock-in by homology-directed repair of the synthetic CAR receptor. They claim that knocking out TCR and MHC will reduce graft versus host disease and reduce rejection of the engrafted T cells for a potentially more durable remission. Their preclinical data supports these claims, and we are still waiting for the results of this trial. What are some of the limitations of CAR-T cell therapy? Antigen escape is one of the more challenging limitations of CAR-T cell therapy and affects a significant portion of patients receiving this therapy. CAR-T cells targeting a single antigen can deliver high response rates initially, but over time there can be partial or complete loss of the target antigen on malignant cells. This is called antigen escape. For example, 70 to 90% of refractory ALL patients show durable response to anti-CD19 CAR-T cell therapy. However, the follow-up data in relapsed CAR-T patients shows a downregulation or frank loss of CD19 antigen in 30 to 70% of cases. Similar findings have also been observed in CAR-T recipients treated for plasma cell myeloma and glioblastoma. Newer treatment strategies focus on targeting multiple antigens using dual CAR constructs, which contain two antigen domains, such as anti-CD19 and CD20, with the hope that this will prolong remission rates. And there are currently several clinical trials looking at this with promising preliminary results. On-target, off-target effects describes the challenges of targeting tumor antigens that are often also expressed on normal tissue at varying degrees, which is more typical of solid tumors. In these cases, antigen selection is crucial to ensure therapeutic efficacy while limiting off-tumor toxicity. One way to address this is to target post-translational modifications in tumor antigens. For example, some solid tumors overexpress truncated O-glycans, which have been associated with poor prognosis and low survival rates in patients across several cancer types, such as gastric, colon, breast, lung, and others. This feature provides a unique enough target to minimize and/or avoid off-target effects. CAR T-cell trafficking and tumor infiltration. Solid tumors have physical barriers that pose additional challenges compared to hematologic malignancies. Direct delivery of CAR T-cells to the tumor, as opposed to systemic administration, attempts to overcome these obstacles. Direct delivery may also reduce on-target, off-tumor toxicities by reducing exposure to normal tissues. This includes intrathecal injections targeting primary brain tumors and metastatic lesions to the brain, or intrapleural injections in cases of mesothelioma, all of which are currently being investigated. The downside to this approach, however, is that it is limited to localized tumor lesions or single foci. Another technique to overcome this limitation is to express chemokine receptors on the CAR T cells that home onto tumor-derived chemokines. Recent studies have demonstrated that CAR T cells overexpressing CXCR1 or CXCR2 showed enhanced trafficking and significantly improved anti-tumor effects. Solid tumors, and to a certain extent hematopoietic malignancies, exert immunosuppressive pressures within the tumor microenvironment. This is facilitated by myeloid-derived suppressor cells, tumor-associated macrophages, and regulatory T cells that produce pro-tumor cytokines, chemokines, and growth factors. It has been hypothesized that weak CAR T cell response, or even CAR T cell exhaustion, is triggered by co-inhibitory pathways generated by these pro-tumor supporting cells. This has prompted investigation into the effects of combining CAR T cells and checkpoint inhibitors, such as PD-1 and PD-L1 inhibitors, to ensure T cell persistence and function. In one study from Children's Hospital of Pennsylvania, 14 children with heavily pretreated B-ALL received a combination of PD-1 blockade and anti-CD19 T cell therapy, resulting in improved persistence of the CAR T cells and better outcomes. Other strategies are looking at engineering, quote, armored T cells to resist TGF-beta-mediated inhibitory signals, or to produce immunostimulatory and pro-inflammatory cytokines to increase survival, proliferation, and anti-tumor activity of the T cells. And finally, CAR T-associated toxicities. Despite having powerful anti-tumor effects, CAR T cell therapies are associated with high toxicity rates and fatalities, and this is why they are not considered first or even second-line therapies. Nearly all patients with acute lymphoblastic leukemia treated with tisagen-lecklusil have some toxic manifestation. The ones I will focus on here are CRS, HLH, and ICANS, none of which have approved prophylactic measures. Recently, there have been studies looking at off switches for CAR T cells. One idea being to incorporate a kill switch that can reduce or eliminate the engineered cells when adverse symptoms take hold. Some of these suicide genes include the use of inducible CRISPR-Cas9 constructs that rapidly deplete engineered T cells. Transient control of CAR-T cells are also under production and investigation as a means to control toxicity without losing the entire therapeutic cell population, and this is seen as a major hurdle in getting CAR-T therapies closer to first-line status. Cytokine release syndrome, or CRS, is a common finding that results from severe cytokine production and massive in vivo T-cell expansion. It can present with mild, vague symptoms like fever, fatigue, diarrhea, headaches, rashes, or myalgia. Or it can be severe with hypotension, shock, respiratory failure, multi-organ failure, and can be potentially fatal. One study quotes 46% of patients treated with tisagenlecleucel for relapsed refractory B-ALL had at least grade 3 CRS or higher. It is believed that CRS is mediated by IL-6, and management focuses on IL-6 receptor blockade using tocilizumab and corticosteroids. Hemophagocytic lymphohistiocytosis, HLH, or macrophage activation syndrome, MAS, occur when CRS is accompanied by a combination of several other factors, such as elevated serum ferritin, increased soluble IL-2, hemophagocytosis, renal failure, and others. The incidence of HLH-MAS secondary to CAR-T cell therapy is unclear due to overlap with high-grade CRS, but is reported in about 1% of CAR-T recipients. HLH can be refractory to IL-6 inhibition and may require chemotherapy instead. Immune effector cell-associated neurotoxicity syndrome, or ICANS, is characterized by elevated cerebrospinal fluid cytokine levels and disruption of the blood-brain barrier. The underlying pathophysiology is not completely understood. Clinical manifestations of ICANS range from confusion, headache, attention deficits, focal neurological deficits, or encephalopathy, all the way up to life-threatening cerebral edema, transient coma, or even seizures. Neurotoxicity following CAR-T cell therapy is relatively common and can occur in up to 67% of patients receiving treatment for leukemia. Management of neurotoxicity focuses on corticosteroids. IL-6 inhibitors are often not effective. So, to summarize, chimeric antigen receptors are modular synthetic receptors that we design to target tumor-specific antigens and activate a T-cell immune response against that tumor. Antigen selection is critical for CAR-T cell function and efficacy. Currently, the most common derivation method involves lentiviral vectors to introduce the synthetic gene into isolated T cells. CRISPR-Cas9 gene editing technology brings new benefits and challenges to the CAR-T cell engineering space. There are still several limitations to CAR-T cell therapy still to overcome, and effective treatment may run the risk of CAR-T associated toxicities, as we have discussed.